Hello, everyone, and welcome to a new ARIMA webinar. We will be rebroadcasting our recorded webinar from last week, and after the webinar, we will open up the floors to myself, my colleague Kai, and also our guest speaker, Argyris Papantonis, for a Q&A live session. So let's get started.
So today we will have two presenters. The first will be a short introduction to 3D genomics from our Director of Research, Kristen Sicking, which will set the stage for our guest speaker, Argyris Papantonis, also known as Akis, who will talk about his lab's pioneering research on the spatial organization of the glioblastoma genomes, highlighting its impact on tumor heterogeneity and providing valuable insights for targeted therapies. Finally, we'll wrap up with a Q&A at the end. So please keep those questions coming for a lively discussion following the presentations. So with that, I'd like to introduce Dr. Kristen Sicking, Director of Research here at ARIMA. Kristen holds a PhD in biology and leads and manages ARIMA's Clinical and Discovery Research Program. Kristen, the floor is yours.
So welcome everyone, and thank you for joining us today. I'm really excited to be here to share some of the latest from ARIMA Genomics. So today I'm going to begin with an introduction to ARIMA's technology and how we're able to explore the 3D structure of the genome before touching on some of the ways that 3D genomics is being applied in cancer research. So I'll talk first about how we can use 3D genomics in concert with other omics approaches to explore the mechanisms of gene regulation. And secondly, we'll discuss how to wield 3D genomics data as a tool to identify structural variants such as gene fusions. And finally, I'll talk about how to integrate the identification of structural variants with information about how that perturbs gene regulation in the local environment around a structural variant. So let's jump right in. To understand the 3D genome, let's start with what we might be a little more familiar with, what we would call linear genomics or a 1G genome. We can define linear genomics as sort of the process of figuring out the order of each of the 3 billion A's, G's, C's, and T's of the human genome in each of our different chromosomes. But of course, there is far more complexity to the genome than can be easily captured in this linear paradigm. What we know now is that DNA is not just randomly shoved into the nucleus of cells. There really is a defined pattern to how chromosomes are packaged into compartments, and this is what 3D genomics is allowing us to describe. 3D genomics goes beyond providing the genetic sequence information. Its real uniqueness lies in its ability to offer additional insights through the spatial dimension of the data. Using 3D genomics, we are essentially able to sequence the structure of the chromosome, enabling us to identify structural changes like gene fusions or indels. Moreover, we can use 3D genomics data to measure the interactions between promoters and enhancers, gaining valuable insights into gene regulation. Now, what I want to start with today is this gene regulatory aspect and talk about how 3D genomics is an important part of a multi-omics approach to better understand the mechanisms underlying gene regulation. Now, in a multi-omics approach, a wide array of tools are available to link the blueprint of the genomic sequence to the various epigenetic factors, such as chromatin state, non-coding sequence modifications, which in turn can exert influence on gene expression or protein abundance or the interactions between small molecules within a cell. And 3D genomics plays a crucial role here in the multi-omics toolkit by identifying the long-range genomic interactions that can orchestrate gene regulation, thereby helping to bridge this gap between the DNA sequence and the epigenomic factors that contribute to changes in gene regulation. ARIMA has developed a number of tools to facilitate investigation of the 3D genome. Our core technology underlying these tools is based on Hi-C, which stands for High Throughput Chromatin Conformation Capture, which was invented in 2009 in Joop Dekker's lab. And at ARIMA, we provide kits and services that allow researchers to query the 3D genome. The product on the left here is what I want to focus on primarily. That is our genome-wide Hi-C analysis, which, as the name implies, looks at the 3D structure of the entire genome. But ARIMA also offers different targeted approaches to achieve reduced representation of genomic research regions of interest, including high CHIP, which uses immunoprecipitation to target interactions of interest, as well as promoter capture high C and custom capture high C, which use sequence-based probe capture to pull down genes or regions of interest to see how those elements organize in 3D chromosomal space. The general workflow of ARIMA's high C technology begins with sample prep using our kits. ARIMA's kits work with a wide variety of sample types and input amounts, including cell lines, frozen tissue, and even FFPE tissue, which is very common for profiling of tumors in cancer research. From a given sample, ARIMA's chemistry preserves the spatial organization of chromosomes within the nucleus by cross-linking the interacting pieces of DNA together. That DNA is then digested, tagged, and proximal fragments are religated to preserve that spatial information prior to NGS library prep. ARIMA's user-friendly workflows are fully compatible with existing NGS library prep kits to easily prepare samples for Illumina short-read sequencing and provide data outputs that can be analyzed using ARIMA's and other NGS analysis pipelines. So now I want to give you a sense of how we actually visualize ARIMA Hi-C data. This is a heat map used by the T2T consortium to scaffold the complete human genome sequence. And what you're seeing here is each of the chromosomes lined up across the top and along the side. Now, each point in this heat map represents physical interactions between two regions of the genome. So the dark red points that you see along the diagonal indicates that there are a lot of these 3D interactions between those regions. And the light red or white indicates minimal 3D interaction. Now, as you can see, it is very common for chromosomes to form these 3D linkages within themselves, but at least in healthy cells, chromosomes rarely form these linkages with other chromosomes. So let's zoom in to look at these interactions in more detail. What we're looking at here is about a 700 kb region on chromosome 2. And to help demonstrate how we would interpret this matrix, let's place 4 points along the interaction matrix here. And what you'll probably notice first looking at this is this bright off-diagonal signal with a high frequency of interactions. And what this represents is that the DNA near the purple point and the DNA at the green point are looping together to give that 3D interaction. To view that another way, we can represent these 4 points in this way along a linear genome. And from the interaction data in the heat map, then we'd expect the purple and the green to be physically brought together. And likewise, the gray and the blue are physically brought together. So we can infer then that the chromosome is organized into domains by these loops that have formed in these regions. So now let's take a look at an example of how Hi-C can be used to gain really important insights into the mechanisms of gene regulation or misregulation in cancer. This example was published a few years ago by Goettingen and colleagues, and they used Hi-C to look at healthy cells versus cancer cells in T-cell acute lymphoblastic leukemia patient. And here what we're looking at is a zoomed in view of the region near the MYC locus, which is indicated here to the left. And nearby is a known superenhancer region. In healthy T cells, we can see that MYC is contained in an interaction domain that is clearly defined here by this sort of triangle and is separate from the domain containing the superenhancer. In TAL, however, that boundary between those two neighboring domains is largely gone, and both the elements exist in a single large domain. Looking at some of the associated multiomics data, we can see that the CTCF binding site near the domain boundary has been lost. And there's also an increase in the H3K27 acetylation signal in the area surrounding the superenhancer. Together, these data suggest that the superenhancer has been turned on and brought into proximity to MYC, leading to a misregulation of the MYC gene, which we also see in the gene expression data. So this is a really nice example of how the 3D DNA organization uncovered by Hi-C informs how MYC is regulated in healthy cells, but also gives really important clues as to how it is misregulated in leukemia cells. Next, I'd like to shift gears to talk more about how we can use 3D genomics as a tool to identify structural variants, including gene fusions. And what really catalyzed our interest and investment into this application of 3D genomics was a collaboration with NYU Langone that began a couple of years ago. They had a pediatric patient, a 14-year-old girl with a stage 2 glioma. And the tumor that she had was resected and analyzed with DNA and RNA sequencing panels, but no driver mutation was found. So the patient underwent standard chemotherapy and monitoring. About 6 months later, however, this patient experienced a very rapid progression, and the tumor returned quite aggressively. So NYU again performed comprehensive DNA and RNA sequencing of the tumor and again failed to identify any driver mutations. So at that point, we performed a Hi-C analysis using ARIMA's Hi-C technology, which revealed a novel structural variant near the PDL1 gene, which encodes a protein that helps immune cells recognize normal, healthy cells in the body. In some cancers, high expression of PDL1 prevents immune cells from recognizing and targeting those cancer cells, allowing the tumor cells to proliferate. So since the Hi-C data implicated a possible role for PDL1 in this patient, our collaborators at NYU decided to look further into that result. And in fact, for this patient, the ARIMA Hi-C result was confirmed with immunohistochemical staining, which showed strong and diffuse PDL1 expression in the tumor cells. So with PDL1 expression confirmed, the patient was then started on immunotherapy with the drug pembrolizumab. And since that change in treatment, the patient has responded very well and her disease is now stable. So what's particularly exciting about this case to me is that by using 3D genomics, we were able to identify a novel structural variant that was missed by these other genomic tests, and which provided the key functional insights that enabled the medical team to prescribe the best treatment for the patient. So now let's take a step back for a second to understand how we're able to use 3D genomics to identify these structural variants in the genome. If we imagine that we sequence a normal cell, we might get a handful of reads covering a gene located on chromosome A and some covering a gene on chromosome B. If those genes happen to be fused in a cancer cell, then with linear genomics, you are relying on the sequenced reads that span that breakpoint to see evidence of the fusion in these two loci. Now in 3D genomics, as we've already seen, we capture information about the 3D structure of the genome. And one really clear characteristic of that structure is that chromosomes most often form these interactions within themselves, but only very rarely between different chromosomes. So in a normal cell, you expect to see a lot of interactions in the region surrounding gene A on chromosome A, and a lot of interactions in gene B on chromosome B, but not many interactions that are spanning that distance. However, when those genes are fused together, as in a cancer cell, now those genes exist on the same fused chromosome, and we should expect to see many reads spanning that breakpoint. So by using this approach, we are able to leverage the 3D information to essentially amplify the fusion signal to detect these gene fusions. And we are able to do so in a way that is agnostic to the partners that are actually involved in that fusion. Now to show you what this signal looks like in real Hi-C data, on the left, I'm showing again a normal cell line, and we see again that the interactions are primarily within the same chromosomes. By contrast, in a cancer cell line, we can see a very distinctive off-diagonal signal. So each one of these squares here is highlighting a 3D linkage or structural variant between chromosomes that is not present in the normal sample. And you can see how that amplification of the fusion signal around a breakpoint results in this very clear pattern that we can easily detect to identify gene fusions and other structural variants. As we've developed this technology, we've done concordant studies to compare ARIMA's Hi-C approach to other gold standard methods for identifying gene fusions, such as RNA panels or FISH. And in all cases in which there are known fusions, we are also able to find that same fusion using Hi-C. This works in a wide variety of solid tumor and liquid tumor types. So we've demonstrated here that we can identify the same gene fusions as existing gold standard methods. But what about other cases like this patient we've already looked at, in which the gold standard tests have failed to find any driver mutations? So is this case that we started with a really unique case, or could 3D genomics be more broadly useful in driver negative cases? So driver negative cancers affect a huge proportion of patients. Almost 500,000 patients in the US alone have tumors with no druggable targets per year. So we've designed experiments to determine whether we could use 3D genomics to identify the druggable targets in cases that were classified as driver negative by other gold standard approaches. We've analyzed over 200 cases to date that were previously characterized with DNA and RNA sequencing and didn't have an identified drug target. In 70 of those cases, we have found with the REMAS technology a fusion event associated with a gene that is a target for an FDA-approved therapy already available today. In another 12 samples, we identified fusions associated with genes targeted by drugs in ongoing clinical trials. And in another 29 samples, we identified biomarkers with diagnostic or prognostic utility. So in total, we've identified biomarkers in 50% of the driver negative samples that could be clinically actionable. Although, as you can see here, the success rate varies depending on the tumor type. We've further tried to validate those results in as many samples as we can using IHC. And in the vast majority of those cases that we've tested, we are actually able to confirm their oncoprotein activity, just like we saw in the PDL1 example. So what we're seeing over and over again with these studies is that 3D genomics can really help cancer researchers in a few key areas. First, 3D genomics can help us detect known fusions that are identified by other gold standard methodologies. But 3D genomics also enables us to find key gene fusions and structural variants that are missed by those other technologies. 3D genomics has also really helped to unlock access to fusions from archived FFPE specimens, which can be very challenging to work with in other molecular assays. Now, in the last section today, I'd like to show one example of how we can integrate these structural variant analyses with the gene regulatory analyses using 3D genomics data. Now, here I'm showing the interaction landscape around a gene ETV1 on chromosome 7 in a normal prostate cell. And next to it is the interaction landscape on chromosome 14 in the neighborhood of another gene, MyPol1. And in the normal cells, these genes have very little interaction between them since they are on two separate chromosomes. Now, if we look in prostate cancer cells, here we see the interactions again on chromosome 7 and the interactions on chromosome 14. And in these cells, there is a fusion event between these two chromosomes. And we actually see the expected interactions between the two chromosomes on the rearranged allele. So in the interaction data here, we can see these looping structures denoted by the circles that are forming between the ETV1 promoter and other regions in an intron of MyPol1 in a region that is also marked by DNA's hypersensitivity. So to prove that these regions were functional, the authors of this study removed the enhancer sequences using CRISPR-Cas9. And they were able to demonstrate a clear reduction of the looping interactions across that breakpoint, as well as a reduced expression of the ETV1 transcript. So as you can see, this is a really exciting case where we can measure directly how that structural rearrangement of the genome can affect the regulation of the genes surrounding that breakpoint. In conclusion, I hope I've helped convince you today that there is much more to the genome than just sequence, and that 3D genomics is a really important part of our multi-omics toolkit. So I've shown you today that 3D genomics is concordant with the current gold standard methods for detecting structural variants, particularly gene fusions, and shows a lot of promise for improving diagnoses in a clinical setting by detecting fusions missed by other technologies. Finally, 3D genomics provides unparalleled access to the sequence, structure, and the regulatory landscape of cancer genomes, revealing novel biomarkers and therapeutic targets. Finally, I'd like to thank the many collaborators who have provided us with samples for some of the studies that I've highlighted today. And thank you all for your attention. I'll turn it back over to Ibrahim to introduce Argyris.
Thank you, Kristen, for that wonderful and excellent presentation. I'd like to introduce Dr. Argyris Papantonis. Argyris studied biology at the University of Athens, where he also obtained his PhD following postdoctoral work with Peter Cook at the University of Oxford. He set up his lab at the Center for Molecular Medicine in Cologne, Germany. Since 2018, he has been a professor for translational epigenetics at the medical faculty of the University of Goettingen, Germany. Argyris, the floor is yours, but I also wanted to mention, thank you so much for posting your questions so far today. And just a reminder, feel free to ask your questions along the way, and we'll get to them at the end. So thank you so much, everyone, for your engagement thus far.
So my lab is generally interested in this relationship between the structure and the function of the human genome. And a way by which one can study this relationship is by perturbing it. So how would you do that? You would choose a system by which this is inherently perturbed and see what the consequence might be. And we do this, for example, as of late, by looking at cells that have escaped the normal route of development, especially for proliferating cells, which is to eventually hit the barrier of senescence and stop dividing, and escape into malignancy. And this would, of course, give rise to a tumor entity and its progression and evolution over time. Now, as an introduction, the work I'm going to discuss today has been pre-printed. So if you scan this code or if you use this DOI, you can find it on the bioRxiv. And it's being almost single-handedly led on the analysis side by Ting Zi, who's a Humboldt postdoctoral fellow in my lab, and wouldn't have been able to happen if Roberto Pallini, who's our neuropathologist from Rome in Italy, had not been generally providing samples and funding for this. So I'm going to be talking about glioblastoma. This is a very common and most aggressive primary brain malignancy. It has, an incredibly poor survival prognosis, and essentially people that have a diagnosis for glioblastoma do not really have a therapeutic solution for them. So we will be using, for this study, we will be almost exclusively using stem cells derived from the tumors, the primary tumors of patients. These are known to be the most resistant compartment of the tumor to therapy, and also be the initiators of tumor recurrence. And what is known to me, for example, as a non-cancer biologist, the sort of hallmark of glioblastoma has always been its incredible transcriptional and genetic heterogeneity. Now, I will introduce our cohort. We have 28 patients, of which some have, from some we have primary tumor material, from others we have relapsed tumor material. For 3 of them, we have pairs of primary relapsed tumor from the same patient, and the tumors emerge at the same position in the brain, and this will become important later. And we also have a funny pair of samples here. They come from a single patient, but the glioblastoma stem cells are derived from 2 different compartments of the tumor, from the central region and the peripheral. So we're going to apply high C to all of them, and the idea, as Kristen already has described, is that you can sort of get 2 birds with 1 stone. So you have a coverage that is enough to give you whole genome coverage, so you can find structural variants and copy number variants for that matter. But you also get the 3D information by which things come together in potentially regulatory interactions in 3D space. And once we've done all our 28 high Cs and we just map them to the reference genome, so this is very important, you need to map to the reference genome, and there's a reason for it, although these people will have really individualized genomes, and we'll get to that. So you map to the reference genome, and then you simply ask how similar are any 2 high C datasets after mapping. And as an aside here, I want to say that, for example, if you have a decent resolution mouse stem cell Hi-C data set and one from a neuron derived from that stem cell, they are pretty close together. They look quite a bit alike if you do these kind of a correlation plot. But here, I think you can all appreciate that there's huge divergence in these Hi-Cs. Even pairs of samples from the primary recurrent tumor, which you could see here marked 275 and 275B, they're closer than others, but they're really not very close together. Now, why is that? This is because genomes from malignant cells are usually riddled with structure and copy number variants that give rise to non-canonical, non-reference structures. And actually was the work from Feng Yue and the University of Chicago that has pioneered our understanding of this, because they have been able, his lab have been able to generate algorithms by which, using machine learning, you can almost automatically identify structural aberrations in these Hi-C maps. And this is usually my favorite example. Whenever you see this sort of butterfly signal, like this one here, on a Hi-C map, this signifies an inversion for which the breakpoints are, perpendicular to the sides of the butterfly. And of course, you can identify everything else, right? A deletion is very obvious. You have a large region without signal. You can find duplications of different kind, and so on and so forth. Now, another thing you can identify in these samples, you can find copy number variants and you can interpret them. And we validated that this is the case by, for one of these samples, we also have whole genome sequencing. And as you can appreciate, we can calculate at the same resolution, which is roughly 10 kb in our hands, we can calculate copy number variation exactly the same way that you would if you had whole genome sequencing. There's very little to be desired on this front. Now, I'm going to show you one example from one of these patients. So this is our line 457. And I think you can appreciate in this Hi-C map that you see all the different chromosomes one after the other, from 1 to chromosome X down there. And you have all this signal off the diagonal. So this is, for example, chromosome 3 here, chromosome 4, and so on and so forth. And Kristen showed you these already. These aberrations in the signal that are really strong as regards the signal intensity, they actually connect different chromosomes in trans, and they represent translocations. And if I make a Serdos plot like this, where you can also see now structural variants within a given chromosome, you can appreciate there's a translocation here, for example, between chromosomes 1 and 9, but there's also a duplication on chromosome 9, and there's an inversion here, and there are multiple deletions and so on and so forth. So there's lots of variants. And if you just repeat the experiment by regrowing the line and having an independent replica, you get exactly the same things with very little variance. Now, how does this look like for our cohort? We actually identify an unexpectedly large number of structural variants. So we have, in the worst case scenario, 26 or 27 of them. In the best case scenario, probably for the patient. And in the worst case scenario for the patient, more than 180 of them. And you can see the dispersion is, so this is a very uneven, pervasive, but non-recurrent distribution. So it's extremely heterogeneous. Inversions are the least represented structural variants, and there's very little recurrence between different patients. So heterogeneity is the key word here. And you can see this here. If we correlate the recurrence, if we try to find common structural variants, you can see that the actual Jacquard index, which means essentially the percentage of overlap of their occurrence, is very, very low. And you can see the highest, for example, are the two lines that are derived from two different parts of the same tumor, the peripheral and the central. And even here, you don't have a convergence that is higher than sort of 45%. Now, if you survey chromosomes and you start asking where would these structural variants occur, we see, and I'm going to start down here, that they very often are clustered. We all need to keep in mind that this is a selection process. So very often, most often, the clones that we get to see are highly selected in the course of evolution of the individual tumors. But here you can see all the structural variants being clustered, and you can see how this looks like on the Hi-C map with multiple events of different intensities occurring together with a copy number increase. So these will not necessarily all occur on the same allele. We don't have this information yet. We don't have allele-specific information. And also, this is a population study, right? So you study essentially the average of many, many cells. But judging from the intensity of these signals, this concerns a large majority of alleles in the population. And you could ask for complex XVs or particular subtypes of them, where do they occur the most? So for example, if you want to talk about duplications, you see that they tend to be in regions where you have GC-rich content, so many genes, and they are in the A compartment, so the active part of the genome, and the expression is generally high there. So you tend to select for duplications that involve active genes, which makes absolute sense. If you want to talk about inversions, for example, there's no bias for expression, and they tend to happen almost equally in gene sparse and in gene dense regions, and so on and so forth. And you can also ask whether these happen closer or further away from topological domain boundaries. So these positions that actually insulate genes and their regulatory elements. And again, you can see that if you're talking about duplications, the breaks tend to be close to the boundary. If you talk about, for example, deletions, they tend to be away from the boundary, deletion of regulatory elements or a gene that sits in the middle of one of these domains. So there's very rich information that you get about the relative position and the contribution of these structural variants to 3D genome organization and its regulatory role. Now, what one could do, and this has been done for other types of hematological cancers, you can actually take the gene expression subtypes of these types of cancer, and there are 3 well-known signatures for a glioblastoma: the classical, the mesenchymal, and the proneural. And what you can do, maybe I should change this, this will be easier. So what you can do is you can actually do RNA sequencing of these tumors. You can classify them in the different categories. And after you do that, we can actually classify 22 out of the 28, not all of them. You can ask whether features, high resolution features of your high C can explain the transcriptional subtypes. And to our surprise, that didn't work very well. So for example, you have some of the mesenchymal here clustering, sorry, these are the classical ones. The classical ones here clustering together, but some other just cluster away. And if you look at even high resolution features, they really get mingled up. So the heterogeneity is such that you can't have a clear picture. So what is going on? This is what we were trying to understand. So what we found is that these structural variants, they provide opportunities to chromatin to form potentially regulatory loops between different parts of the genome. And there are 2 pretty large surprises here. So I'll give you an example. So this is the HLA locus. You have 3 active HLA genes, and you have a deletion in between these 2, the G and the J. And very often, in actually 5 out of our 28 patients, you actually have the formation of a neoloop. You can see one example here, another one there. You can see the loop forming there, connecting the promoters of these 2 genes and activating them both. Now, if I look at neoloop formation throughout the cohort, you can see that some have really high numbers of neoloops forming, more than 1,000, and some have very few, in the order of few tens or hundreds. And what is fascinating is that all of the opportunity arises in hundreds of cases in all of them because of hundreds of structural variants that we can map. The existence, the emergence of a structural variant does not necessarily mean that a loop will form. So you also need compatible, in some manner, regulatory elements and genes on either side, for example, of a deletion, so that they can come together. So there is scope of mapping these. And one part of the scope is that you can uncover new predictive markers. And this is the HLA-G gene that I was talking about, which has until today not been associated with glioblastoma, has been linked to some extent with melanoma. So you can take this gene and ask in the TCGA cohort of hundreds of glioblastoma patients, how does it look like? Well, it is on average overexpressed over normal tissue. This overexpression is not because of copy number variation. You can see the copy number variation here between the high and the low patients, but patients that have a high expression of this gene, presumably because of this neoloop forming, actually have a poorer survival prediction. And overall, we have more than 6,000 of these neoloops to look into. Another type of neoloop would be the obvious one, connecting the promoter of a gene like IFIT, shown here, with enhancer elements in its vicinity. So here what we have is we have an inversion that brings these enhancers in proximity to the promoter and allows the formation of these two neoloops that turn on the expression of this gene, specifically in a couple of these patients, but not in the other ones that do not have such a structural variant. So this means that very often what Kristen presented as enhancer hijacking can actually take place. So the question is, how informative is this, right? How often does it occur? And we can actually see that the fraction of loops that explain such events is really higher for neoloops than for normal loops pre-existing in these genomes, or for enhancer loops that will eventually, even if you don't map that, of course, talk to a particular gene, which is not the case, for example, for promoter loops. And this, most of the times, is linked to a hyperactivation of these genes. I should have a small parenthesis here and say that there's been a very nice work from Feng Yue published a few months ago, where some of these loops can actually be classified as enhancer repressor loops. So instead of turning on a gene, which is what you would normally expect, the classical enhancer hijacking paradigm, you can have a repressor hijacking to turn off genes in the vicinity of these structural variants. Now I'm going to give you another example. Here's the ADAM9 gene, again, never really associated to glioblastoma biology. You can form, there's neoloops forming in its vicinity, two of them with a promoter, one that sort of encompasses and forms a new gene locus. This is because of the deletion here. And you can see that, again, if we go to the TCGA data, ADAM9 can be found significantly overexpressed in the cohort. It's not because of copy number variation. And you can actually see this poorer survival also being associated with it. So the point I'm trying to make here is that these neoloops that one cannot assess by having linear type of genomics, like whole genome sequencing, can be important. You can see here GO-term analysis of genes linked to neoloops that are either enhancer promoter or promoter. And you can clearly see, for example, that glioblastoma signaling is significantly in genes relevant for glioblastoma signaling are significantly enriched in this particular category. And what I'm trying to get to is that neoloops should be co-considered when we're trying to understand the biology of these tumors. And I'll give you two very obvious examples. So one is CDKN2A. This is the famous P21 protein. Very often, these locus is deleted because this allows cells to escape senescence and cell cycle arrest. And you can see that in most of our patients, there are actually 13 behind this color here. We have a deletion in the P21 locus, but we have a neoloop formation in the P21 locus in 4 cases. And in these 4 cases, this also leads to the repression of P21, presumably via repressor promoter of P21 interaction. Now, another hallmark gene is EGFR. So many of these tumors are driven by overexpressed EGFR. And you can see here that the amplification of this locus is also a very highly occurring event. In our cohort, we have 9 GSC lines that are amplified for EGFR. But we also have another 4 that are actually, sorry, this was 3 here, and it's 4 here. We have another 4 that actually have an enhancer promoter contact that forms de novo, so a neoloop because of a structure variant that can turn on EGFR. This is all nice and good, and it's good for the biology understanding of these tumors and generally the evolution of the tumors. But are there chances that one can use Hi-C diagnostically to inform therapy, like in the example that Kristen described previously? Now, we have 1 patient out of the 28 that showed really high expression of MYC. And we looked into this patient. So this is the glioblastoma stem cell line 148. And what we found is that there's a translocation between chromosome 12 and chromosome 8, linking the MYC locus, a very small region with a 1 megabase region carrying multiple really highly expressed enhancers. And you can see the formation of two rather specific neoloops. Not everything contacts the promoter. So these enhancers here and that enhancer there make a neoloop with MYC. And actually, to make this even more interesting, this whole translocation is then amplified, presumably as extra chromosomal DNA in this particular patient about 30 times. This means that we have a huge overexpression in just a single patient of MYC, a gene that is, of course, a known driver of carcinogenesis, but not often involved in these types of tumors. But nowadays, there are inhibitors that one can use to target MYC, and there's one, EN4, which is a very recent development and can actually interfere with the phase separation properties of this particular transcription factor. So we can hit our patient that has the MYC amplification and translocation with this inhibitor, or a patient that doesn't carry that. So 62 here, and you can see the difference in how we are reducing the amount of positive MYC positive cells here. We have a significant effect in the ability of these cells to keep proliferating, and we have some increase in the amount of cells that are led to cell death. So we wanted to explore this a bit more, and we thought, is there a way of in silico modeling this interaction and trying to understand it? So what is the minimal? And I'm not going to go into excruciating detail here, but there was a very nice interface for modeling chromatin folding in 3D and structural variants by Mario Nicodemi's group a couple of years ago, and this is called Prisma. The problem with this for us was that this was not designed to find to make predictions and modeling in trans, so for a translocation, but only things that happen in cis, which are slightly easier to model. So Mariano, a physicist in our group, he actually took the task of making this work. And what he managed to do is not only make it work, but also make it have some predictive value. So again, I will not go into the details of the physical modeling here, but what we can have is we can have models that nicely recapitulate the 3D interaction that we see in our high-C data. They do this just by using two pieces of information, the K27 acetylation and the RNA-seq that we have for this one locus. We have as a negative control another locus where the translocation doesn't exist. We artificially fuse it, and as you can see, we cannot get neoloops here because we don't have the propensity heretofore. And then you can actually look at the simulations behind these reconstructed 3D maps from the modeling. And each one of these is essentially a single allele, or if you want, a single cell. And what Mariano found in this is that the interactions of MYC with any of the two neoloops, or with any of the two enhancers, is actually mutually exclusive. He could never find a situation. So here is MYC on chromosome 8, white, in this hub here with, you know, the large enhancer, the really broad one, and here it is chromosome 8 again in white, chromosome 12 in black. They come together, and you can see here the single enhancer in light blue. And this was, you know, a bit surprising. So Mariano took to the task of treating this as a single cell experiment and predicting the outcome. And you can see here in panel F what it would look like if we were to plot it like a single cell experiment. Very often in gray, you wouldn't have a contact because, of course, that's part of the game. But very often you would have this mutually exclusive contacts with either one or the other enhancer. And there are now, you know, well-established ways of making prediction of how the gene will burst into transcription on the basis of these contacts. And I think you can appreciate that there's very different patterns occurring in the different simulations, which could actually explain the vast transcriptional heterogeneity for making this particular case in this type of tumor. And now, as a last bit, I would like to discuss this comparison between the primary and the relapsed tumor. As I said at the beginning, we have three pairs where we have derived glioblastoma stem cells from the primary and the relapsed tumor from the same patient. And these relapses were always occurred at the same position inside the brain. And what we find is, and this is not unexpected, of course, is that we have more structural variants in the relapse situation. This makes absolute sense because, of course, these patients have undergone heavy chemotherapy and radiotherapy. They've all undergone the exact same therapy, by the way. And of course, this will induce lots of DNA damage, presumably, and provide more opportunities for structural variants to arise. However, the prevailing theory that local recurrence of glioblastoma is essentially a continuation of the previous tumor cannot be justified by our data. Now, I'll give you a very simple example. You can see, for example, here, there are some amplifications that you don't find in the relapsed tumor. And let's say that this extrachromosomal DNA might get lost. There are here 3 very apparent strong deletions, 2 of which completely disappear from this data. It is impossible to lose a deletion and recreate the DNA that you've lost. So these and other such type of observations when comparing the 2 landscapes using Hi-C allowed us to predict that these are likely, they have a common ancestor, most likely, and it's difficult to assess that, but clearly they are not one the evolution of the other. The most likely scenario is that the primary tumor is removed by the initial wave of chemo and radiotherapy, and this gives an opportunity to the relapsed tumor to develop under the pressure of chemo and radiotherapy and provide a rather different new clone. And of course, if you look at the gene expression profile, the difference between the 2, you can find genes that explain the transition and the aggressiveness of the relapsed tumor. And then if you use the Hi-C, you can find that enhancer promoter contacts actually justify in all 3 pairs the majority of differentially expressed genes. And of them, the gain of enhancer promoter neoloops is what seems to make the difference for the activation of these genes. So I would like to stop here and conclude sort of 4 basic things that is at least our take-home message from this study, which is the first such large-scale cancer study in my group. So we can leverage Hi-C to generate patient-specific scaffolds of chromosomes and interactions. And when we do this in glioblastoma, we discover remarkable heterogeneity, both as regards structural and copy number variants. And what is really astonishing to me is that there's very little recurrence of most of these, the vast majority of these. The opportunity that these structural variants and copy number variants provide is that they allow patient chromosomes to give rise to neodomains and neoloops that seem to be specific for the most part for individual patients. And these can now not only explain transcriptional heterogeneity, but they can actually be used to uncover new dependencies over the course of time. So this is more or less it. I'm happy to discuss any aspect of this and also give you more details about the analysis in the back. And thanks again for taking the time to listen to me.
So let's open now the floor for questions, and then we bring also Argyris Papantonis, our guest speaker, into the discussion. The first question we have here, Argyris, very interesting talk about the structural variants in glioblastoma. Do you think the same frequency of SBS will be present in pediatric glioblastoma, and how does it compare with the frequency in other cancers?
So we have not looked ourselves into pediatric gliomas, but there is a paper from the Saratz's lab in Science Advances from a couple of years back, and the extent of structural variants is not as great as the one we see in adult glioblastoma. It's quite a bit, but it's not as much. I think from what we're seeing, because we've done similar work on pancreatic cancer, and there's data out there from a number of different cell lines, a couple of tumors, and epididymoma, which were direct patient samples. I think glioblastoma by far exceeds any of these in the number of structural variants one identifies with Hi-C.
Another question we have here is how many cells are required for each of these Hi-C experiments, and why did you not use the full patient samples rather than introducing more noise by random sampling for single cell probes?
So one could, in theory, use tumors. This material is far more difficult to get your hands on, especially to get your hands on it at a quality that will give you good data. We are actually currently testing 4 tumors that match 4 of our lines with different numbers of structural variants, hoping to get enough data. But the problem, of course, is that you will have an even more heterogeneous population to work with, because the tumors also contain non-tumor cells from the brain, and it depends on how this tumor looked like and how well the surgeon managed to remove enough of a piece to be able to do it. Typically, we use, so you measure, according to the ARIMA kit, which is what we use, you need about 1 million cells to get a feel of how much chromatin you can get out of these, and then actually try and do the experiment. For these particular glioma stem cell lines, it has been very difficult because they are very idiosyncratic. We don't have 2 lines that are the same. So on average, we use about 1 million cells for one Hi-C experiment. Some required less, so you had to go to 500,000 to get good data. Some required more because they were lysing very, very poorly, despite our better efforts. So it is not as easy, and even the amount of playing around you need to do with individual samples actually makes it very, very difficult to look at these and trying to do the tumor, because with a tumor, well, whatever is left of the tumor after all the pathologists look at it and so on and so forth, you essentially have one shot and that's it. So this is one of the reasons why.
Thanks, Argyris. Just also to add some additional information, our recommendations, as you mentioned, with the ARIMA Hi-C kit is 1 to 2 million cells, but we have already produced high quality data with as little as 50,000 cells. Yes. We also have some researchers that have used the kit with 6,000 isolated nuclei. So you really can bring down the number of samples if you don't have, it is really precious.
Yes, we have never tried that low, and to be honest, I would be scared to do this on a precious tumor sample. Yeah.
Another question we have here is that I think the resolution of the Hi-C experiment is lower than micro-C experiments. Do you have any plan to do other micro-C, or do you think that the resolution with the Hi-C is enough?
Yeah, well, we're doing micro-C for other projects, and it does work very, very well. That is true. And if you really want to focus on enhancer promoter interactions, and this is your focus, then I think that micro-C would be a very nice solution. Again, the question is, can you really apply micro-C to some of these samples? We don't know yet. We are trying, but not for structural variants. I think for structural variants, this is more than enough. And we've probably, in this case, over-sequenced just to find the variants.
There was a question about the particular package that we use for analyzing the SBS with the Hi-C data. I don't know if you, Argyris, have used the tools that we recommend at ARIMA Genomics, or you have your own tools?
No, we use directly the suite from Feng-Yu's lab. So the combination of NeoLoop Finder, Break Finder, Eagle-C, and so on and so forth. But I think they're very, very close to one another.
Yeah, I mean, at ARIMA, we recommend a combination of different tools. Basically, we use HiCap to align the Illumina reads, and then the alignment pass through the Hi-C break finder, which is what we use for the SBS calling. Then we also integrate that with the user or a QC of the Hi-C data, and also to overlay it on top of the Hi-C map using JuiceBox just to visualize the data. Still open problem is the normalization of Hi-C data originating from genomes with copy number alterations, while matrix balancing approaches should partially cancel out the imbalances in total Hi-C signal orienting from any locus. The resulting locus distortions in the interaction matrix are not completely corrected.
Yeah, so this is a good point, and actually this has been now addressed. So you can't do it with matrix balancing, it's not good enough. There's actually CMV-based balancing, so you detect the CMVs on the basis of using the linear information in the Hi-C data, and then you use that in a combination of matrix balancing and normalizing to get rid of them. If you look at our preprint, it's described in the first supplemental figure exactly how this is done, and there's an example visualizing the result. It actually works very well, but you need to correct for this.
A new question: is chromatin structure stable or dynamically change, particularly for disease-associated genomic regions?
Well, I think it's absolutely dynamic, and what we're capturing is a small proportion of these configurations and an average along a great population. I mean, in order to do this properly, you would need to have also imaging, live cell imaging to look at whether, enhancers and promoters come together and how often. But, this is a limitation of all these genomics-based 3D genomics assays, and it is such.
And are there any sequence or epigenetic feature associated with the stability of change regions?
Well, on average, the most stably associating regions in genomes, in high C maps in general, are CTCF loops, and they appear as this very distinct dots. Having said that, there've been live cell imaging experiments that sort of translate that into a time course, and actually these loops are in this looped formation, I don't know, between 2 or 3 minutes, and then they spend the rest half an hour being unlooped. So, it gives you an order of magnitude of what happens. Can I just jump here and say that Kai has sort of copy-pasted to me a question from Jane Meller. So hi to Oxford, Jane. There's indeed a famous Nature paper from the Benson lab looking at glioblastoma, and they have done high C. The difference between that paper and ours is, well, first of all, there were no tools back then to actually do structural variant calling, so that was impossible. And they deliberately used in that paper IDH mutant tumors, which means that there was a methylation defect that took away lots of CTCF bound positions, so distorted the insulation patterns along chromosomes, and that was the big finding. So you didn't have insulation, and you could have loops forming across boundaries. Ours have been deliberately chosen to be IDH wild type, so that whichever neoloops we detect are not because of hypermethylation of CTCF binding sites and eviction of the bound CTCF. So there's a difference there. I don't think we see the particular neoloop that they saw, also because this gene is not a driver in the IDH wild type gliomas, as far as I know.
Okay, I think we have a time for maybe 2 more questions. One is, if I understood correctly, using RNA sequencing, you were able to differentiate between the different groups of glioblastoma. However, with high C, you did not differentiate, samples were mixed. Is the structural variant supposed to affect gene regulation? How can you describe this observation?
Yeah. So that's the whole idea, right? That the structural variants diversify enough 3D genome organization that allow for deregulation of genes, because they now talk to or don't talk to the regulatory elements anymore, or talk to new regulatory elements, and these can be almost equally enhancers or silencers. And we actually looked into this because people that have looked, so it was again Fenyu's lab, when they looked into leukemia samples, they could actually stratify the subtypes just based on high C. And we thought, can we do the same? And it's clear that we cannot do the same in the case of glioblastoma. It's actually pretty mixed. It's not absolutely mixed, but it's mixed to an extent that you can't just say all the mesenchymal and all the non-mesenchymal will just cluster away on the basis of 3D genome structure. They're just too heterogeneous.
Okay, maybe we just have the last question is, how about the reproducibility and coverage of chromatin interactions by high C experiments? What is your experience about?
Yeah, so we've, it's actually incredibly good. So if you just make a couple of replicates of completely different growing cultures of the same GSCs, or in other cases of similar cell types, for example, senescent cells or whatever you want, and you correlate the structures that you get from a good high C experiment, they're really, really highly correlated. And there was, I think, a plot that I was showing at the very beginning of the talk where the correlation between replicas was really, really high, and everything else was essentially not so.
Also look for DNA methylation signatures in your samples?
We have the data, but the analysis is not done, so I can't really say. But yes, we have, and we're also now trying to see if the high C can stratify on the basis of the DNA methylation signatures. There's a question at the very top that I want to address. So Georgi says, the simplest explanation for your relapse is that you're sampling different cells each time from your cell line generation. Now, that is not true. The relapse GSCs and the primary tumor GSCs are two very different populations, and they've been grown out of the tumor the exact same way. So I guess the question is, of course, we're not sampling the whole tumor, right? So I'm careful to say that this is an observation done only on the basis of the GSC. But it is pretty clear that you can't just explain one directly from the other. And it's also pretty clear, and this was my big surprise when I looked at these cells the first time, that if you look down the microscope, they look very different. They look incredibly different. So you could have one line forming a spheroid and another one that grows adherent and looks very, very mesenchymal with protrusions and everything. So it's really not the explanation. I think the full explanation is not that they are two completely different cells. They have a clear common ancestor. You see it in their patterns and in their structural variant signature, but one is not an evolution of the other. So the relapse is not an evolution of the first one. It either represents an underrepresented clone in the tumor or a different evolutionary path that started early, was dormant, and there's quite a few people now that favor this idea of dormant GSCs that wake up when they're given an opportunity.
I think Argyris, there is another question related to the classification of the genes involved in the structural variants. You affect many different genes in different patients. And get different structural variants. But do these genes belong to the same pathway? How else could one explain that you get the same disease with such heterogeneity?
Well, you get the same disease in the sense that you get a tumor of largely unknown origin in the brain. And it has some, general phenotypic characteristics. So many of these tumors are, EGFR, hyper-expressing EGFR. Fine. We do see some convergence. So there are some pathways that tend to be commonly affected, right? You have an anti-senescence pathway. So the regulation of senescence is affected, but this has to be, because if you don't, turn that switch off, you won't be able to evade cell proliferation. So there are some things that are common, but you wouldn't say that, it's a, the MEX signaling, for example, and then you just hit different genes along a defined signaling pathway. We don't really see that. And this is, this is, I guess, what makes the tumor also very difficult to deal with. So I see a question from someone that works with plants and said, I don't work in humans, I work in plants. I don't think the variation would be so much mainly due to the presence of long introns, so the structural variants don't make much difference. Yeah. So of course, I think this needs to be clarified. Of course, you have many of these SVs happening in positions of the genome where either they have no effect, they can cut a large gene in two that wasn't really that much expressed and that doesn't really affect the physiology of the cell, or you can have it in a gene desert, or you can even have it at the boundary between two topological domains, but the resulting fused domain doesn't result in changes in gene expression. All these things are plausible scenario. We have examples of all of them in these different tumors. And I think some of these are just, co-occurring. So you have one that is selected because it has an effect. It co-occurred with another chromosome or nearby, and they are co-selected, even if this structure variant doesn't really have a functional outcome. It's absolutely reasonable, and it happens a lot.
Okay, I think last one that just entered, you mentioned the methylation data is still being analyzed. Do you have also data on histone modifications, albeit these are mostly adult patients?
So we have data on K27 acetylation, and we have some CTCF binding, not for all of these tumors, for some. And nothing else, nothing more than that. Our focus was always at the beginning to look at enhancer promoter interactions.
Is there a way to see how representative your neoloops are in the whole tumor? A single cell spatial DNA analysis, the world is too full of meaningless cell line work.
Yeah, I mean, we can discuss how meaningless cell line work is when the cells are derived directly from tumors, but I do see the point. No, single cell high C will not give you these types of information, unfortunately. So one would, in an ideal scenario, you could identify subsets of cells in the tumor that, somehow you identify that they are alike, and then you take these and do a sort of low cell input high C on all of them and try and see how many of them have the neo-loop that you're most interested in. And I think this, this could be a solution. However, there is one thing that I think needs to be highlighted. For the vast majority of the neo-loops we discussed, not for all of them, but for the vast majority, the signal in the high C map of the formation of the neo-loop is pretty strong. So it belongs to the really high quantiles of the overall contact signal in our maps. This signifies that it represents a large proportion of the cells, probably not all of them. So at least within the heterogeneity of the sample that we have, we are looking at a significant proportion of cells. Now, it is clear that, for many of them that are weak, we don't know how many cells we're talking about. Is it 1%, 5%? There are computational ways of assessing that. I know that ARIMA has done a couple of tests in silico for this, and, they claim that, so Kristen the other day was saying that, down to 2 to 5%, you can already see things. So we might even be capturing very rare events that might be reasonable. Sorry. And the translocation ones, as a follow-up, yes, these, well, you wouldn't see the neo-loop, but you could look at the translocation with traditional, whole genome sequence. So you could find that the translocation that we're detecting is true. That's pretty easy. Or with a fish experiment, that of course one could do, but you don't have to do it because you're sequencing the same genome anyway. So now that we're going to get the first tumor samples, I'm sure that the translocations will be definitely the same. There's no doubt. The question is, what fraction of cells will they be represented in? And that will be slightly more difficult to calculate.
If we were not able to get to your questions, I think someone from ARIMA Genomics will reach out to you guys in the next days. So yeah, I thank you everyone for attending the webinar. Thanks Aki so much for the amazing talk, and also to be present here in the Q&A live session. And yeah, I wish you everyone a nice day, and cheers.
Thank you very much.